Hello everyone, myself Vidika Dilip Gardi from TYBSC. Today we are going to talk about a post-translational modification process called as protein phosphorylation. So firstly, what is post-translational modification or PTM? PTMs are nothing but the modification which occur onto the protein after they have been synthesized. So, as here the name phosphorylation suggests addition of a phosphate. Here, after the protein has been synthesized, phosphate groups are added to specific amino acid residues on proteins. It is a very common regulation method in the body and plays an important role in process of cell signal transduction, gene expression and differentiation. So, this is a reversible mechanism which is carried out by enzymes called as protein kinases, wherein phosphate group is added to the polar R group of various amino acids. So, now how these proteins are phosphorylated? So, first is kinase substrate interaction. First, it is necessary to have this both interactions so that they could react and phosphorylate the amino acid. So, first is the specific target proteins, which are nothing but our amino acids, are recognized by our protein kinases, wherein kinase substrate complexes form. Then, this kinases phosphorylate specific amino acids such as serine, threonine, and tyrosine. But this choice of phosphorylation site is influenced by the amino acid sequence which is present around it, and it can definitely be regulated by various factors. Now, as the kinase substrate have interacted form a complex, now how does it phosphorylate? The kinase enzyme takes the phosphate group from the gamma position of ATP and transfer it to the hydroxyl group of target amino acid residue. So, as this transfer introduces a negative charge, it alters the conformation of protein and often its activity. So, as you can see in the picture, the gamma position of phosphate is this now transferred to the target molecule, which is our amino acid. Then, protein phosphorylation and cancer. So, as you all know, that there are various regulatory mechanisms in a cell or a body which are necessary for the functioning of the body of our body. So, if there are any mutations, deletions, defects in it, definitely some conditions will be seen phenotypically in our body. In same, protein phosphorylation is also involved in regulation of multiple biological processes and over expression of kinase. So, if there is mutation or a defect in this regulatory mechanism, it would lead to activation or dysregulation of kinase pathways, which will be the base for multiple tumors. For example, here is given example of chronic myeloid leukemia, wherein there occurs chromosomal translocation. So, due to which this kinase enzyme is stuck on its own position, which will go on phosphorylating the amino acids. This will lead to the proliferation of various tumor cells. So, there are chances of increasing of cancer. So there is also another study wherein oncogene VSRC is used. Here the tyrosine kinase has a phosphate group of TYR527 which has a role in tumor cell proliferation. So the studies of oncogene is extensively used for Rous sarcoma virus, which is the main cause of sarcoma in chicken. Also, kinases aberration have various types of cancers. For example, there is amplification of HER, which is observed in breast cancer. So, by doing all these studies, doing all this research, we can make use of targeted drug therapies. So if we know where are the mutations or the deletions present, targeted drug therapies can be made according to that. So it would inhibit or repress the action of this kinases. So there are various methods or techniques by which we can study protein phosphorylation. First is mass spectrometry. Mass spectrometry is a analytical technique wherein mass to charge ratio is used to identify the um, sample. It's it's a powerful technique for identifying and quantifying phosphorylated proteins and their sites. Next is a Western blotting. Western blotting is mainly used for protein recognition. So here antibodies, which are that phosphate group specific antibodies, are made so that they would recognize the particular phosphorylation events on the specific proteins. Third is kinase assays. Just as we do enzyme assays, wherein enzyme substrate complexes are seen, enzyme activity of how of when adding substrate would be seen. So this in this kinases assay, this kinase enzyme along with the substrate are incubated, wherein radiolabeled ATP is present. So to measure this kinase activity, we can use this. Fourth is phosphatase activity assay. So we know that phosphatase breaks down the phosphate group. If there are proteins, amino acids which are phosphorylated, this phosphatase will dephosphorylate this amino acids, and its activity we can measure using colorimetric or fluorometric methods. This is also same as enzyme assay, wherein our activity can be measured. So here are the list of references which I have used to refer my topic. Thank you. Hello everyone, myself Vidya.